So we are here at the Massachusetts General Hospital Cancer Center. Various questions in cancer biology try to be answered here, but what our role is to bring proteomics into this game. So proteomics now especially with multiplexing proteomics and bringing the whole technology to high throughput, we are really able to answer very complex questions where it just takes to look at many samples. So TMT multiplexing, it's just what it allows us right now is doing 10 samples, looking at 10 samples simultaneously. The technology has been out for a while. It has been out for 10 years now. For us, and this was when I still was in the Giga Lab as a postdoc, it really took off when HCD allowed us to use this on an Orbitrap. They have this method now which they call the Simultaneous precursor selection MS3 method, which is further improvement of our MS3 approach. And what this allows us is to really use isobaric labeling, use TMT at a high throughput with very great accuracy. And so this whole combination is really helping us pushing this stuff through right now, getting this accurate data at high throughput, to look at those many samples and really play on the same level as genomics was. One aspect that plays a very important role in cancer biology is a deregulation of phosphorylation events. The phosphorylation is there to transduce signals throughout the cells. It's very important to look at these phosphorylation events to understand what is going on there. And this is something we do. We go in there, we look large scale for phosphorylation events and see how they change in the cancer environment. And we not only do this now between one normal tissue versus a cancer tissue, now we can do this with a high throughput method between hundreds of samples and really can feel out what are the common mechanisms that go on that are deregulated there. Right now what we're doing, within 36 hours, we are comparing 10 full proteomes, which means we're already under 5 hours that we need for a full proteome. The nice thing about using multiplexing right now is that we can increase the throughput at 2 different layers. One is happening on the mass spec side. At the other side, we bring in another layer by saying, okay, we can put in higher throughput through more multiplex channels. And so we're bringing this both together, which really gives a big push in the throughput. The Orbitrap Fusion is a tribrid mass spectrometer, and it combines 3 kinds of mass specs, which is a quadrupole at the very beginning, then the Orbitrap itself, and an ion trap. For us, this constellation within this mass spec is very important because this is what allows us to do those MS3 scans, which is not possible with another instrument. It gives us this possibility at a very high speed. As I just told you, we can do these 10 proteome comparisons within 36 hours. And this is only possible because we have the Orbitrap Fusion. 95% of the work we do in the lab is based on this technology. One question about this, and this is especially about the MS3 method, is the sensitivity. Can we really use this for all kinds of samples, including post-translational modifications, or including pull-down samples, IP samples, that we need to check for the interacting protein of biomolecules. What I'm dreaming of is to really, it's very similar to genomics, to increase the throughput of this technology, thereby bringing down the price. And I'm thinking of, it will take another year, another 5 years maybe, to bring down the price for a full proteome to $100. I think this price level then, this is one of the arguments that will come very good when we say we want to use this in a clinical setting.